And Philip, it's actually our most senior student of the second round of PhD students at the UAC. And he is not only past his halftime, but really right now preparing his defense. He will be defending in the end of September. And he is actually looking for opportunities. So if anybody out there is listening either now or watching the record later and has a opportunity where you think the skills and the background of Philip could fit, then I'm very happy. I'm very sure that he will be happy that you reach out with those opportunities. And without further ado, I will now, Philip, open the digital stage to you so you can start presenting. And you are going to talk to us about Florence, which is Florence labeling in recoded E. coli with non-canonical chemical entities, which I think is a pretty um, scientific title, but I'm sure that you're going to explain all of it to us about what it means. Thank you so much, Philip, for being with us and looking forward to learn with you.
Thank you very much, Eva, for this kind talk, and I'm absolutely super happy to be here and presenting the work that I've been doing in the past 4 years. I just have a technical question. Can everyone see my presentation in presenter mode?
Yes. Correct.
Right. Okay, cool. Right. So in the next half an hour or so, I'll basically talk about methodologies in doing single molecule tracking and why this is important. So in essence, what I'll be talking about are not really fundamental questions, but more on how can we build a system to study useful properties of molecules. I will briefly mention the protein translation process, which is kind of the core of where antibiotics are targeted in that context. And that's actually the bread and butter of my supervisor's lab. I will talk about the single molecule tracking methodology and why is that important and what sort of information we can get. And then I will dive into more on what I did as a PhD student. That is, I'll introduce the genetic code expansion and why is this a hot method to ride this wave. What have we been developing in terms of bacterial strains, in terms of vectors for protein expression, some dyes and natural amino or the chemical entities, as we will see forward. And finally, we will look at some nice applications on how powerful this methodology actually is. So to start with, protein translation that is common for all prokaryotes and eukaryotes, the focus here now is in bacteria. And it's a pretty complex process. So it's pretty central to life. It's very synchronized. So you don't have to pay too much attention to this figure over here. The whole point here is that there are a lot of players. And within a living cell, for a long time, it has been impossible to know how they behave in real space and real time. But they're very important because each of these steps can be targeted with different antibiotics. And each of these steps kind of has been designed by life in such a way that it can raise resistance to all of these antibiotics. Most of the behavior or the data that comes out on how fast these processes are comes from a test tube. But what Magnus's lab has been doing in the past couple of years is figuring out how all of these processes actually behave in living cells. So they have been able to decipher the initiation process in living cells. They have been able to decipher how ribosomes behave in living cells, that is, how fast these processes are. But what is still an open question that I will touch a bit upon is the elongation process within this particular part over here. Now, if we take a step back, what I'm essentially saying is I'm quoting George Gershwin, who says that basically life is like jazz. It's best when you improvise. And although all of these processes look kind of improvised, they're actually highly synchronized. So you can think about the protein translation essentially like a jazz band, where each of the jazz players have their own role. And like if you listen to jazz, that's kind of a bit of, I would say, non-harmonious, or yeah, it doesn't sound like a classical defined way as you would have with pop or anything like that. But this is basically how this works. Now, when we talk about single molecule tracking, so because we want to do this in living cells, single molecule tracking is a optical fluorescence microscopy. And historically, it comes down from following the planets or the celestial bodies in the sky. So basically, what people did is they used telescopes. I mean, Galileo, Copernicus, all of these guys. But when you look at the movement of big bodies, biologists and physicists figure out that at the same way you can look at the movement of small bodies within the cells. And each of these individual components is a protein. Now, what I like to kind of show it this way on especially what is the single molecule tracking all about is that if you take the Starry Night picture from Van Gogh, you have a reference point here, which is the planet Venus, and you have the Aries constellation, which is over here. So Earth is moving relative to stars. Stars are, in a way, also moving or not. But the whole point is if you're able to decipher the coordinates of these spots over here, you would be able to determine when this picture was painted. And actually, historians, by using the location and the position of the stars relative to Venus, have been able to pinpoint an exact date of 1886, summertime, for when Starry Night was pictured. So that's what you can do if you just track the movement of particles. These guys, in this case, are pretty big particles. But then if you look at the Aries constellation, that and you compare it to a fluorescently labeled molecules in a living cell, they kind of look pretty much the same. It's just the size or the scale that that is different. Okay, so this is me, and I would like to show how molecules basically move. So each molecule is an individual, like all of us, and within a given state, this molecule moves around. Now, I spend most of my time in BMC, bound to my bench and my lab. So this is one of my states of living, so to say. But then, like a couple of years ago, it's Uppsala, we like to drive bikes. I was driving my bike pretty fast, and I ended up in a state that was pretty slow. So I broke my arm. And basically, all of a sudden, my movement or my state of being became quite slower. And I ended up with a cast. But that being said, so I was actually still able to pipe it. Anyway, the whole point here is that a molecule is an individual, and we somehow need to put a label. So we somehow need to know where this molecule is. And what single molecule tracking is telling you about is the identification of a molecule. So with mathematical modeling, that we're not going to go into the details, we're basically able to extract the ID card of a molecule in a living cell. That being said, we're looking at these three crucial parameters, and that is the diffusion coefficient, which is basically something quite unique to each of these states, and you can sort of think of it as a personal number or personal number. And when a molecule is freely moving around, it has quite high value. When a molecule is bound to something or it's kind of stuck in a way, like I was with the cast, it has a very low value. Then we're talking about the state occupancy. Occupancy is basically what percentage of these individual molecules are found in a different state. So for example, for me, slow state 1 at BMC is a pretty much highly occupied state. In the bike, I'm not much of a biker, so that will be kind of lower occupied state. And basically the mathematics behind that also gives you dwell time. That is time spent in a given state. So how long we are found in this state. And that's basically the basic information you're getting out from single molecule tracking. Now, the lab has successfully labeled proteins and tracked them by methods of electroporation, where you basically pre-label your molecule of interest, and you use electricity to get it down into cells. You grow it on agarose pads, and you get the personal identity of the behavior of that molecule. And this works very nicely for nucleic acids, but for proteins is pretty much limited to the protein size and charge. Or you can use so-called self-labeling tags, where basically you label molecules in the cell. This has been shown on a couple of instances, so works pretty robust. So what you have is that the protein is expressed with this label. You add a color or a dye, you wash it out, and basically you have the same behavior experimental wise, similar as you would do it with the electroporation. However, the problem here is you cannot put this dye wherever you want. You're kind of limited to the structure of the protein, but also these tags can be problematic for very small or very dynamic proteins. And that's where this genetic code expansion, or GC, comes in, which is a technology that uses the translational system of the cell. You have amino acid, or a chemical entity, as we like to call it, which is made by organic chemists, so that's purely designed. You have something which is called a delivery system, or an orthogonal translation system, that puts this chemical entity within a protein structure at an exactly defined position. And this defined position comes from the genetic code. So the genetic code is redundant, which means that it encodes for different components or different amino acids, but there is a codon which is blank, or three codons which are blank, and then they do not encode for anything. Now, if you think about it, this, from labeling perspective, is great because you have minimal protein perturbation. So compared to a big labeling tag, you are not going to influence much of the protein structure like we see over here. But also, at the same time, you can install it site-specifically at different position to get different information. And again, this is done in situ. So you don't have to electroporate. You don't have to pre-label. You let the cell use its own translation machinery to install this chemical entity that we put it in. Now, that being said, the problem is that this stop codon is recognized by a release factor, which most of the time would outcompete the chemical entity on the stop codon, and you would end up with proteins that are just don't have the chemical entity, which is a problem. And that's why engineers or bioengineers have generated this bacterial strain that remove the this release factor 1, so that now you have a strain where these chemical entities or natural amino acids can basically enter. Now, the way we want to do this again is we have the chemical entity with this delivery system to the ribosome, and chemists have developed something which is called click chemistry. And click chemistry are basically sets of reactions that are non-toxic, that are ultra fast, and they are pretty much specific. So they don't react with any other components in the cell. And one such example is this particular chemistry over here, where you can vary the side chains of the molecules. But if you install two very specific groups, these very specific groups will recognize themselves in space and time, and they will connect the two molecules. And this was such a cool technique that back in 2022, it got the Nobel Prize. And even the lab, Magnus's lab, by doing this sort of protein labeling, made it in the public on how this technology can be used to label proteins in living cells. So this is a video done with the constructs by one of the colleagues, Linnea, and Ivan, of course, he helped quite a lot with that. So that being said, we already have a couple of components that we have to think about. Now, when it comes to microscopy, genetic code expansion depends on these chemical entities. And this is how these chemical entities look like. All of these groups are reactive and can react with a partner. So these are recognized by the ribosomes. They get into the proteins and they react with different dyes. But what we have recently found out is that these particular groups, sort of that were known in literature but underexplored, can still be used for robust labeling. So the whole idea here is that you have your molecule A with this reactive species, and you have your molecule B or your tag with this reactive species. These reactive species recognize each other, but they do not interact with other components. Okay, so what has been such a big caveat so far is that for E. coli, this system has been underused. Now, I was talking about molecules that are moving fast, and the only report so far in E. coli has been done by the Johann Elf lab, where they have taken a intracellular protein that is inside the cell. They have tried to label it, but they were pretty much unsuccessful because there was no specificity of the chemical entities that they were using back in the days. Luckily for them, they were able to label molecules that are on the cell surface. So you can see here, for example, if you apply the chemical entities where you don't have this stop codon or blank codon, you can't see anything. Once you have the chemical entities with a stop codon, you see very nicely on the membrane. But for getting chemical entities inside the cell has been a challenge, and that has been underexplored. And the only way for single molecule tracking that people have done again is using it in mammalian cells. But again, in mammalian cells, they're always targeting these receptor molecules simply because the chemical entities back then, so we're talking about 2016, 2020, haven't been really there. So now with the technology development, we have something more that I will show a bit later. But the whole point here is that you see all of these images are basically images of molecules that are on the cell surface and not inside the cell. And that is crucial because Magnus's lab, they like to study protein translation, and all the protein translation happens inside the cells. It doesn't happen outside the cells. And one of the problems of using a normal E. coli is that the Johann Elf lab found out that if you use a normal E. coli for click labeling with all of the stop codons, you basically get some sort of toxicity. So what do we need? So with this in mind, we have the necessity to develop a method that can allow us to label proteins inside the cells, in this case, inside E. coli, but at the same time, it should not be toxic. And that's why there is a group of bacteria which are called recoded strains, we are looking for recoded strains that have doubling times similar to the parent strain, so the wild type that we use in the lab. They have the same doubling times, which basically means you have the same protein translation, you have the same self-morphology, and if you just switch one codon from the table that I'll show later on, you're not going to get misincorporation. We're looking for orthogonal translation system that is very specific for the chemical entities, that by itself is non-toxic and is high efficient. And we're looking for chemical entities, which are basically dyes and non-natural amino acids that give you very high signal, and you can get easily rid of them from the cell. So the recoded strains that were originally made at Harvard University, they have doubling time of 60 to 80 minutes, and a normal E. coli has a doubling time 30 minutes, and they're very unattractive for physiological studies because basically they're pretty much sick. So a recoded strain, or this C321, is basically a strain where the stop codon has been removed. So all of a sudden, you have two stop codons in this strain. You have the UAA codon, the UGA codon. You can make all the other amino acids, but there is no UAG codon inside. That being said, you can generate an orthogonal translation system that recognizes this UAG, and you can basically put in whatever chemical entity you want background free. So we were optimizing this strain by doing genome editing, by repairing some DNA repair genes, which we got first generation of strains, that is GRE1, repairing some metabolic genes, and repairing part of the translation machinery. So we generated 3 strains in this past 4 years, which was a huge challenge for us. And basically what we're seeing here is that compared to the wild type, so starting from this recoded strain 1, if we do a little bit of fixing, we get quite nice effect in terms of the fitness. And then finally, we are somewhat close to the MG1655. That being said, we can also look how these bacteria look on the plate. So this is basically our recoded strain, the original one from Harvard, and this is one of our final strains. So this strain looks pretty much the same in terms of what we see with a normal E. coli. And now if you look at the doubling times, if we look at the last parent of our final working strain, which we call GRE3, we basically have doubling times that are similar to MG1655. That being said, by doing rational genome engineering or fixing of these strains, if you say we can or we have generated strain that is similar to our wild type. And this is how basically this looks on in terms of growth. So you can put it on a mother machine or a chip, and you see that all of these strains divide very uniformly. There is a very nice colony size. There's a very nice cell size overall on the mother machine on how they grow. So this is just basically a way to look how cells grow. However, if you take the original strain from Harvard and you put it in here, you see that the colonies are very non-uniform. You get this elongated phenotype, which means that this strain cannot grow at 37. So in essence, by just doing a little bit of engineering in that sort of way, we can get to a natural-like phenotype for this recoded strain. Okay, now we switch gears, and again, we move to the vector. So we have created a reporter which has the stop codon that can now respond to any orthogonal translation system in any chemical entity. And for a long time, we were stuck with this vector 1. And what I'm saying here, and this was in a way sort of painful to work with, because this vector 1 in our particular strain, for some reason, didn't respond. So you can see down here, if you take the fluorescence or the signal of the reporter. So first of all, if you don't add a natural amino acid or a chemical entity, and if you add the chemical entity in this instance, you're basically getting something that looks like the background. But once you do a change in your vector, and that took us also a long time to figure it out, you all of a sudden get a very nice fluorescent signal, meaning that the reporter is active, meaning that this orthogonal translation system recognizes this stop codon, it puts in the chemical entity, and then you get a very nice signal over here. And so how does this look under the microscope? Basically, this is the background of one of the recoded strains that we will work with. So in the channel for the reporter, we see quite a nice background. In the channel for the dye, we see sort of background. If we now add the dye, we see similar behavior like our background. That means the dye can be washed. One of these chemical entities can be washed. If we add the unnatural amino acid again for a stop codon, we all of a sudden see that our reporter responds, which means that the reporter is being produced. The orthogonal translation system recognizes the amino acid, and we are getting the reporter. And finally, if we add the dye, we get increase in signal. That means that the chemical entity is incorporated at the particular position we want to have in. That means that the dye recognizes the chemical entity, and that means that all of them are pretty much specific. And this is just a control where we, for a codon that is not recognized by the orthogonal translation system, we add the dye, we add the natural amino acid, and we basically something that looks like the background. So In our case, we are able to wash, we are able to produce proteins, and that's actually pretty cool. Now, what is actually very important, and for people who like to work with this kind of stuff, is that the dyes or their properties change. The chemical entities have different properties depending on the reaction partners they react with. That being said, this particular dye has one sort of signal when it's by itself. It has one sort of signal when it reacts with a partner non-chemical entity, and it has another sort of signal with a partner, which is another chemical entity. So that being said, you have to be very careful when you choose the chemical entities and the dyes. And this has been really underexplored in literature. And another thing that we kind of struggled quite a lot with the chemical entities is that over time, when we put things under the microscope, depending on these partners, we were losing the signal. So what is this graph telling you is that there is a chemical reaction that undergoes that, in principle, you get high fluorescence value over here, but over time the fluorescence degrades and you end up with signal that is basically dark. So again, this was a bit of a painful process and we were scratching our head around it. But then just by changing the chemical entities, we were able to fix this. Now, finally, we were able to validate this on a microfluidic chip, which was the initial proposal on the thesis that I was working with. So this microfluidic chip are manufactured in the Johan Elf lab, and they have published quite a lot on this. So the way this works is that you have this sort of lanes where you put in labeled bacteria, the bacteria are growing out, are dividing. And the nice thing is that, first of all, you can put in antibiotics in a synchronized way, meaning that all of these bacteria will get the antibiotic quite equally. And number two, from an experimental point of view, it's very user-friendly for data acquisition, because this non-canonical or chemical entity labeling is not as efficient as some of the methods that have existed inside the group. So you can imagine that you can get a lot of data and you can finish up your experiments quite fast if you run this on the mother machine. So as a control to identify the states or to assign personal numbers to these molecules, we took a protein that the group has already characterized, and that's a membrane protein, and we have labeled it with a halo label and with a click label. And we are expecting this one to be pretty much membrane bound and to be pretty slow because it only moves on the membrane and it doesn't do essentially anything. On the other hand, we have an intracellular protein, so protein inside the cells that we label with two different techniques that we are basically comparing. So what are we comparing here is the data for a halo dye and a click dye. So for the leg-Y click labeling, once we put it on the mother machine, we can clearly see these dots over here. I just need to move my pointer and show how this looks on the mother machine. So the click labeling basically is there. Each of these dots are individual molecules and they are diffusing, some of them like this one. For example, if you look like into the center, this one pretty slow. So this is what we're expecting to see in terms of the movement of this molecule, free diffusion around the membrane. And that looks the same if you put in the second label. So the dots are pretty much immobile. I mean, they move, they wiggle left and right simply because that's how they behave. But this is, in terms now of numbers, a very good or a very big relief for us. In terms of the personal number of the diffusion coefficients, we get comparable diffusion coefficients in the slow state. Let me just put in my pointer back. In the slow state, we're getting somewhat different diffusion coefficients in the fast state, but that's simply because currently we have a lot of data that we're processing and optimizing from a computational perspective. And because this is a molecule that doesn't really diffuse around, we can see that the occupancy for the slow state is for the halo label. Let's see now. Right. So the occupancies are okay. This is just a bit of a hiccup. So this is the click label, and this is the halo label. So it's only like a problem with the color, but basically, these two occupancies in the slow state, regardless of the labeling method, are similar. And in the fast state, so you're comparing these two in the fast state, we have similar values. So that means that if you take a protein and you label it with two different approaches, and you get relatively the same numbers, you validate your method that it works. Now, what about the fast state? If we just take an intracellular protein that diffuses fast and we do the same labeling approach over here, we label it with click chemistry, and we label it with the halo label. So here we see that those molecules are moving pretty much fast. They're diffusing pretty much fast, and they look kind of similar in that sense, regardless of the labeling approach that you use. And again, here we see that basically we don't see any diffusion coefficients for the slow state. We see very nicely somewhat correlated diffusion coefficients for the fast state. But what are we basically showing here is that regardless of the label, this intracellularly labeled protein that doesn't bind to anything occupies most of the time fast state. These are just some artifacts that we're currently working on, but that's only a computational bottleneck. However, with this sort of data, and this is like for us pretty exciting, we can now assign fast state and slow state with two different labels for this particular method, which hasn't been shown so far. And finally, just to give you a pinch on what I will be kind of writing about and where we are, is that we are looking for measuring the binding time of one of these factors that I mentioned previously in the presentation, and that is the elongation factor G. And you will hear about it more from Adrian's project, which is kind of correlated to this. But basically, what we know is that we do not know how fast this moves, and we do not. And literature basically shows us, and experience from other research groups is that we cannot label this with a halo tag or any fluorescent protein. And that's where the natural amino acids come in. So some of our preliminary data, it's a bit puzzling in this sense, but we're working on it, especially now with the microfluidics, is that most of this EFG has a 30% occupancy in the slow state and 70% occupancy in the fast state. That means that it's moving there. However, I will take these numbers with a big pinch of salt, because we're now reworking or reevaluating on how we're going to track this particular molecule. And this is just a video of how a single labeled EFG looks like. So it's moving pretty fast. This is inside the cells. And yeah, we can label it, we can track it. We're just now optimizing certain things. And you will read about this more into the thesis. So with this, I would like to conclude that we have generated a recoded strain with parent-like properties for single molecule tracking, which is very attractive for the imaging community. We have vectors, or one of the vectors, for proper expression of this orthogonal translation system. One very important take-home message is that choose your chemical entities very carefully. The photophysical properties depend quite a lot on each other. We can use genetic code expansion to track molecules on a microfluidics chip. And we have a robust experimental setup platform that can be used for testing antibiotics for people in research. And we can label, we can track EFG. We're just working our way around it with this new kind of setup. With that, I would like to thank the Johansson Lab and especially Magnus for giving me the opportunity to work on this pretty interdisciplinary project. Anneli, who's been instrumental in working on the EFG project, Ivan, who takes care of the lasers, and Eric, who's been fiddling around with the pipeline. I mean, he's been actually doing quite a lot of help in the analysis part, which are definitely not trivial. And of course, I would have to acknowledge my colleague Linnea, who was working on the GC labeling. Then Gerrit Brandis from the Brandis lab and Suparna Sanyal from the Sanyal lab. Gerrit has been instrumental in working on the recoded strain and all of these genome engineering, and Suparna Sanyal for the discussions on the EFG that we are kind of finishing around. And they, of course, have been very supportive towards my career overall. And of course, the ELF Lab members, especially Jimmy, who's been instrumental in teaching the microfluidics, and of course, Spartak, who is been very instrumental in doing the analysis on the microfluidics. And of course, the people who pay my salary, and that is the Uppsala Antibiotic Center, but also colleagues from the Uppsala Antibiotic Center, first of all, for the great times that I have had in these 4 years. And here I have to point out Adrian, who's been also helping quite a lot with the discussions for the EFG, which is related to his expertise. And with that, I think I'm done, and I'm happy to take any questions.
Thank you so much, Philip. So great. Let's see if you're getting some applauses over here. So just so you know, let's see if someone might have a question. They can put their hand up to open up the mic or write it down. Meanwhile, just to tell you, Philip, what a great work you've done. And I know there's been a lot of troubleshooting, and working to optimize a big method like this is a lot of work. But I'm sure that the community will be very thankful to you and your group for a really long time once this is published, and other people can also start to use it. So I hope that you can feel proud yourself of the work that you've done.
Well, it has been, well, I have to point out that it has been a team effort, and we're very lucky to have Magnus and his vision that has been pushing this all around. And I think one of the important things here is the vision that we've been very much privileged to have, like with Magnus and in that sort of way.
Meanwhile, if other people have questions, can you go to the slide where you were showing actually that you show how the validation of your method, that you were showing how the molecules are moving, fast state, low state.
Yeah, exactly.
So actually, that reminded me a little bit of, I don't know if you guys had back in the days when there was no streaming, these channels that were coded and you needed to have a decodifier, and then you had to pay for those channels. It looked exactly like that. So I wonder, because, I mean, what I understand from the mother machine is that the mother machine, those bacteria are actually growing and moving along the channel.
Yes.
So what kind of time scale are we talking here that it doesn't seem like the cells are moving at all?
Yeah. So here's the thing. All of this work for single molecule tracking is done on agarose pads. Now, for example, the way I look the mother machine is like multiple agarose pads, very nicely organized with a very defined position where I can get a lot of data out of it. Because to get sort of this analysis to say, or to make a solid claim, you need a lot of data. The problem with the click labeling is that once you do this on a agarose pad, you're very much limited to you have to take like 100 or 200 positions of cells that are just like randomly lying around. And from an experimental point of view, that's very tedious. The mother machine is a very elegant way where you have everything packed and it's all there, number one. And number two, you can easily buy the media. Like if we go, for example, here, by changing the media, right, on this cartoon, by changing the media, you can apply antibiotics, and you know that all of these cells within all of these traps are getting the antibiotic more or less the same time. So we're not really in this point, it's more of a technical advancement with further, of course, scientific advancement of what people can think. So now basically the imagination is the limit. You have a system, the system is relatively robust. think what you want with what you can do with it.
Great. Yeah, I was wondering also like if the cells are moving, but I guess the molecules are moving faster to what the cells are growing. So then
Yes,
you have that difference
yes,
in time. All right, understand. Um, if nobody has a question, we do need to take a short break because we're supposed to start with Carla's talk at
sure,
15 past, but then we'll take a 5 minute break. Of course, I know, Philip, you'll be happy to take any questions privately via email if anybody wants to reach
absolutely.
out. Thank you so much, Philip, for your presentation. I wish you the best in this last push to the
Yeah,
defense, and I will be there at the defense celebrating with you.
absolutely. Thank you so
Okay,
much.
everybody, let's take